to welcome Dr. Kishore Bidasee to the University of Trinidad and Tobago. To some of us here, Professor Narang Singh, Dr. Maraj, Dr. Baladin, and I, he is a favorite student and classmate. So I can't tell you how proud I am. You know, I've known Kishore since he was in first year. But why I'm proud, and I'm putting an onus on him, but I know he can shoulder it, is because you will be able to see firsthand the class of degree that we turned out from the University of the West Indies, chemistry department. Okay? The Dr. Ramrup Singh and Dr. Varma, they too can take pride. All right? So anybody who thinks that a homegrown degree from a tertiary institution in Trinidad and Tobago is substandard, I introduce you to Dr. Kishore Bidasee, who went from beginning to end in the chemistry department at UWI, from start to finish. And as he himself told me very recently, when you have a good foundation, you can go anywhere. So I present you with someone to whom we gave a good foundation. Well, so don't go anywhere.
Well, thanks a lot, Dr. Stout, for that wonderful introduction. It's really, really a pleasure for me to actually come and speak here. This is probably the first time since I graduated from UWI that I am actually speaking, in such a, forum. So basically, I am a chemist turned pharmacologist. What happened is that when I was in the chemistry department, a lot of individuals used to be actually isolating chemicals and putting it on the ledge after they're done, analyzing it. And then I wanted to actually take those chemicals and see what it can do to biological systems. So that's basically chemistry, but just making a little bit of a change. Now, since this is a very intimate audience, if you have any questions whatsoever, feel free to stop and ask. I speak very fast, but very understandably. Okay, so don't worry about that. I can, handle all my slides. Oh, I just thought, you would have seen the number of slides I have here, but don't worry about that. So what we want to do, since I am a chemist turned pharmacologist, is to develop new drugs. And the disease that I'm very much interested in is a disease that is very, very close to a lot of people in Trinidad, which is diabetes mellitus. Okay, so just in a nutshell, I just came back, Dr. Stout from Stockholm, and she asked me to give the same talk that I gave in Stockholm, but I think that would not have been an appropriate talk, because what happened is I assume that the audience is at this level, and then I just move on to what it is. So what I wanted to do here is basically boil down the disease I'm talking about in a nutshell. Okay? So diabetes mellitus is basically a syndrome that is characterized by high glucose levels in the blood. About 350 million people worldwide have diabetes mellitus, and we are anticipating by 2025 to have about 500 million people with diabetes mellitus. In America, about 10% of the population have diabetes, and I'm sure in Trinidad we almost have about 15 to 20% of the population either have frank diabetes or have the metabolic syndrome, of which 50% of these individuals will end up getting diabetes mellitus. Again, in a nutshell, diabetes mellitus arises from a defect in the production and secretion of insulin by the pancreas, or the inability of the peripheral organs to respond to the insulin. So either the pancreas is not making the insulin, or the peripheral organs such as skeletal muscle is not actually responding to the insulin. So that is basically what causes the high levels of blood glucose. And this is a very simple schematic. I like to present my work in schematic form because I think it is very, very easy to understand. So there are just a few players in the pancreas. What we have are the glucose transporter. We have the next channel called the potassium ATPase channel. We have a calcium channel, and then we have the insulin, which is inside these cells in vesicles. And what happens is when blood glucose levels rise, this glucose binds to the glucose transporter, and then it gets into the cell, but that metabolism of glucose actually changes the ATP-ADP ratio inside the cell. Okay, so again, what I'm doing here is explaining chemistry, but from a biological perspective. Once that ATP level go up, then that ATP then binds to this potassium channel, it closes the channel, and then the cell becomes depolarized, calcium enters into the cell, and then fuses with the vesicles to cause insulin to be released. So again, that is basically the nuts and bolts of how this pancreas operates. Now, a lot of interest worldwide is on trying to find ways to increase insulin secretion in individuals who have at least type 2 diabetes. In individuals who have type 1 diabetes, there is no insulin being made because the beta cells of the pancreas, they are shot dead. Okay? So again, in a very, very simple form, what we have is insulin being made by the beta cells. Once we have insulin release as a result of a meal or so, then that insulin will cause glucose to be taken up by the skeletal muscle, and then it shuts down glucose production in the liver. So what the liver does is actually make glucose. So if we have a high levels of glucose in the circulation, glucose production in the liver is shut down, and then what you basically have is glucose being taken up by the organs and then we lower blood glucose levels. If the pancreas is not working, then we have type 1 diabetes. And if this peripheral system is not working very well, we have type 2 diabetes. Now, these are the two major types of diabetes. There's also diabetes that occur in about 5% of pregnancies, referred to as gestational diabetes. My niece, who is pregnant right now, she told me she has gestational diabetes. And then there are certain drugs that can also cause diabetes. And this is, like for example, drugs that are used to lower, let's say, diuretics. These water pills, they also elevate blood glucose levels. So this is the nuts and bolts of what I'm going to be talking about. Individuals who have diabetes mellitus, right now, they develop a whole bunch of end organ complications. And these are things that you are quite familiar with. Kidney failure, stroke, lower limb amputation, impotency, blindness, nervous system damage, heart failure, periodontal diseases. I'm just listing, quite a few of these complications that actually occur. And then, of course, one that is very close to our heart in Trinidad is cognitive deficits. And the reason why I say in Trinidad is because I notice a lot of the politicians in Trinidad, they basically, have some, they're going small, but they end up very big. So I'm thinking if you tell them they have cognitive deficits, then that's going to cause a little bit of a problem. But I'm sure there's something, related to that with the politics in Trinidad. Anyhow, so cognitive deficits in type 1 diabetes results in a whole bunch of various, issues. Cognitive deficits in type 2 diabetes also results in a whole bunch of, various, problems. Okay? Now, the reason why I separated it into the type 1 and type 2 is because the etiology of type 1 diabetes is a little bit different from the etiology of type 2 diabetes. Cognitive deficits, as I mentioned earlier on, this is one of the newer problems associated with diabetes. The ones that I talked about earlier on, they are well known. Okay? This is what your heart would look like if you have diabetes. This is just an animal model. So this is what a normal heart would look like. It's contracting, it's relaxing. So this here is when the heart is filling with blood. Right here is when the heart is actually contracting. Normal heart. And in this animal model of diabetes, after 12 weeks of diabetes, this is what the heart looks like. So instead of the heart contracting like this, it'll just be, okay? And it's very, very progressive. So I'm just showing you some, images that you can actually relate to with respect to, diabetes and heart failure. So we use an array of techniques actually to get into more details, but I don't think it's so important, here today. The management of diabetes mellitus right now involves glucose-lowering agents such as insulin analogs. If you have type 2 diabetes, they start you off on metformin, then you may progress to sulfonylureas. You have exercise, diet management, and of late, there's a lot of information, educational information, to try and educate the population about what to eat, what not to eat, etc., with respect to this diabetes mellitus. But like I mentioned earlier on, this glucose lowering strategies that are currently involved are insufficient to effectively manage these end organ complications, because even though we have a whole bunch of glucose-lowering drugs on the market, individuals with diabetes mellitus still end up with a lot of complications. So that means that these drugs are not really working. So there's an urgent need to identify strategies that can be used in combination with glucose-lowering approaches to prevent these end organ complications. Now, this is a question that I was, when I was in a graduate school, where do we start to find these adjunctive strategies? Okay, so this is where you as a graduate student will have to, start doing your research work. Where do we start? So what we know, when I did a little quick scan of the literature, we know that the severity of the end organ complications. Now, I mentioned earlier on, diabetes causes many organs to fail. And the only thing that actually link all of these various organs is the blood supply. Okay? So there's the severity of the end organ complications is strongly correlated with the degree of microvascular dysfunction. So then for most of you here, what is the microvascular? So again, these are very, very simple questions that you ask, but by asking these questions, you can really understand the system you are dealing with. So the microvasculature, basically, in a nutshell, is the blood vessels within an organ. So you might have, let's say you have your heart here, you have your eyes here, you have your brain. So within each of the organ is what you have the microvasculature. The vessels that connect the organs is referred to as the macrovasculature. Okay, so we are not really interested in the macro, we are interested in the micro. And then the microvasculature basically have, on this side we have the capillaries, and on the other side, the blue, we have the arteries. But in the capillary section, what we have is the endothelial lining, and we also have a little bit of smooth muscle cell surrounding it. Now, why we need the smooth muscle cell is because the smooth muscle cell helps to contract the vessel. So if you want to regulate blood pressure, then you don't need to contract your vessels. What you need to do is to dilate your vessels if you have hypertension. So a lot of the drugs that are used to regulate blood pressure actually is moving the blood vessels from being like this to just being like that. Okay? And the way how it works is through the smooth muscle cells, causing them to relax. Any questions? Am I speaking too fast? Is it difficult to understand? Okay, so in 1954, a grand old gentleman at the University of Nebraska Medical Center proposed this free radical theory of aging. So when I moved to Nebraska, this guy was like 94 years and he was just like this. But he was, plugging along really, really well. And he developed this theory of aging. And basically what this theory said is that in healthy individuals, you have a balance between the oxidants and the antioxidants, okay, within the body. Now, I'm not going to go into any details, but that's basically the gist of what he said. And then in diseases, many, many diseases, what you have is more oxidants within the body and then less antioxidants. Okay? And this happens in the case of diabetes mellitus as well. But what happened at the same time was that clinical trials after clinical trials using antioxidants failed. Okay? So they thought it was actually the oxidative stress that was involved, but when they tried these clinical trials, when they performed these clinical trials, all of them failed. And this is one that I really like, because they had tried up to 1,027 different strategies to treat stroke in individuals with diabetes mellitus up until, this is, I think, is 2012. And every one of them basically failed the whole process. So that when clinical trials fail, people have a lot of reasons why it failed, right? So they went into this with a lot of promise, but it failed. What they basically said is that they went into these patients too late. The antioxidants was not the best in the world. Maybe the animal models they used to get these drugs or these strategies were not good, etc., etc. So that is, and then this one guy basically said, despite decades of research, it is currently impossible to state if exogenous antioxidants in fruits and vegetables can really promote health. Okay, so everybody started to, question themselves, are they really looking for the right thing, or are they, just going after an epiphenomena, like I had mentioned, up there. So at that time, I was actually working in the area of cardiac pharmacology. So basically, I do a lot of cardiac work. And what I was trying to understand is why in the diabetic heart we have more Christmas lights than in control heart. That was a very, very basic thing that I was trying to understand. So we had published a few papers in that particular area there, and then we started to observe that when we look at the protein levels of the... Okay, yeah, I think, so basically, okay, let me back up a little bit. Good question. So the heart is actually, a pump, right? So if you, in order for it to contract, you have to stimulate it. And this is evoked. So what we did in this particular experiment, we isolated the cardiac myocytes, and then we have them in a dish, and we have some electrical current, and we can then stimulate them to mimic exactly what is going on in the heart. So if you have a myocyte from a control animal, when you stimulate it, basically what you see is a very nice rapid release of calcium in the cardiac myocyte, and then it dies off. I apologize for not explaining this in more detail. So when you stimulate it, it go up, calcium go up in the cell, it comes down, and when calcium go up in the cell, then that cell contracts. If calcium doesn't go up in the cell, that cell will not contract. Okay? And then when you have these Christmas lights, this is the fundamental basis for ventricular arrhythmias. Okay? Christmas lights. So what happens is this cardiac myocyte, you cannot have calcium being, available in a cell. Actually, the way how they categorize calcium, it's like a menace to the cell. So it's always locked up. Okay? If it is released, it's going to do something. If it's locked up, then it's not going to do anything. So there's a whole system of, like storage units in the cell that will prevent that calcium from just wandering off. Okay, so calcium is the primary metal in the cell that is responsible for a lot of, events. Really good question. Okay, so what I get excited about is, and a lot of people don't get excited about, is this little, funny shape. Because this is telling me exactly what is going on and stuff like that. Okay? Really good question. So again, feel free to stop me anytime you don't understand something, and I'll explain it, to you. So basically what we were doing is we had isolated then the major proteins that were involved in this process. So basically here is calcium release, and this is calcium uptake. And in the cardiac myocyte, there are just a few proteins that are responsible for these phenomena. Okay? So one of the proteins that is, in fact, the primary protein responsible for calcium release is a protein called the ryanodine receptor. And believe it or not, this particular receptor was isolated because of a compound found in Trinidad, ryanine. And when I was in Trinidad, I never heard of this compound ryanodine, and I was living right here in Trinidad. Okay? But
now for cardiac muscle, you have the storage of calcium. It's a little different than other muscles. Did you look at that?
Yeah, so the storage of calcium is actually the endoplasmic reticulum or the sarcoplasmic reticulum. Right. Yeah, so basically if you... Yeah. Right. So the storage of calcium, what we are more interested in right now for this particular...
That had been screwed up as well.
Yeah. But I think, other people had looked at it and there wasn't any major problem. So in addition to the ryanodine receptor, and this is circa here.
These ones that you're looking at are true for all muscles.
Yeah, every cell type have a version of the ryanodine receptor or
but
a
you didn't
version.
look at the storage.
What's that?
You figured that you thought the storage, because the previous work did not affect this at all.
Really, really good question. So the storage is actually...
Because that's what's unique to cardiovascular muscle.
Right. The storage, the proteins that bind calcium, let's say calciquestrin, and there's one other protein I can't recall right now, right? But they didn't, have any change in function. We had, done that. So this ryanodine receptor is, if you have a bank, then you will have an entry door, you will have an exit door. So the exit door is the ryanodine receptor. Calcium comes out from the storage like he was talking about. And then in order to go into the bank, calcium uses a protein called SERCA to go back in. And every cell have a version of the ryanodine receptor or a version of SERCA. Okay, with the exception of red blood cells. Now in biology, what we have is a lot of redundancy. We have a lot of isoforms. Okay? So I study, like the 3 different types of ryanodine receptor, the 3 different types of SERCA, those sort of things. Good question. Okay, so what we are finding is that the protein level wasn't changing. This is in the cardiac muscle, but the function of the protein was actually changing. And we asked and basically, going back to chemistry, what I did was a very, very simple experiment. So you don't have to do rocket science to figure out things. So way back in 2003, what I was doing is isolating these same calcium storage units, and I was doing fluorescence scans. And what I found was that in the control animal, the SR vesicles, there was no fluorescence. But in the diabetic vesicle, there was fluorescence. And then sort of like a light bulb went out in my student and my head. And then we realized that when we talk about oxidative stress, everybody talk about reactive oxygen species. But there's a next group of oxidants that is present in the cell called reactive carbonyl species. Okay, so this is a little bit different from the reactive oxygen species. And what these reactive carbonyl species are small mono or dicarbonyl species that are synthesized from glucose and lipid metabolism. And what these things do is, so then we did a quick scan. So even though we had the ryanodine receptor and SERCA protein that did not have any change in levels, when we start to probe for all of these reactive carbonyl species, we found that in every case, we have a lot of these reactive carbonyl species increased in these 2 proteins. So again, this is a little bit tricky because what is happening is that when you buy antibodies to do Western blot, you buy antibodies for specific proteins. But then we were looking at antibodies not for the protein, but for these adducts that were commercially available. Okay, so what we now have is a protein that is extensively modified by these reactive carbonyl species. And then we had a whole bunch of papers that actually showed that these modifications will impair in the function of the protein. So the protein we study, for example, the ryanodine receptor, is a very, very big protein, 2.5 million Daltons in size. Okay? So what we had to do is we had to cut up that protein and determine exactly where the modification was located, and then do a whole bunch of experiments to determine, to mutate the protein, and just to show that the modification was actually impairing the function of the protein. Okay? Any questions there? Uh-huh. Uh-huh. I'll come to that a little bit. I'll come to that a little bit later, because so we were most of these here are the endogenous, electrophiles. So we didn't want to, use models. Yeah, so what we did is, and I'll show you that a little bit later on. Really good question. Uh-huh.
A lot of the insulin mimetics we use right now, just as Dr. Professor Nagasing said, they tend to use dipicolinic acid,
Mm-hmm.
and that is a naturally occurring metabolite.
Mm-hmm.
So basically, you're trying to make a cure with something with a poison. Is that something?
Not really. I'll talk about the biochemistry of this a little bit later on. First of all, you have to identify what is the problem. Okay? And in this particular case here, what I was just looking at are these endogenously generated reactive carbonyl species. Okay? And then the compound you were talking about, that particular compound actually is not in clinical trials. And the reason why I say it's not in clinical trials, or it's not, further along, is because when you are actually trying to test a compound in clinical trials, you really have to understand side effects. Because in vitro, you may have a compound or so working, but then when you have to put it in the animal, you have to check renal function, liver function, cognitive behavior, etc., to make sure that that compound is actually not causing any additional burden on the patient or so. Okay, and I'm going to get to that a little bit later on.
Let me ask you a question now, because You brought up in the beginning with the antioxidants that the animal models were bad, right? So the antioxidants did work on dogs or whatever animal model it was. How does this help? Does this give you any clues why those animal models?
Let me just go back to, if you have a minute here. So basically the question he was asking is this here. So after you lost a game, all right, you want to go back to the drawing board to figure out what was wrong.
Right.
So these are some of the things that actually people thought was wrong. The animal models, the biomarker.
Okay, so that's just false.
Yeah, they don't know. They really don't know. And actually, after a clinical trial fail and you're out, let's say $300 million, you want to, start firing people. So you have to come up with, excuses or at least something to justify why it is, this thing failed. Right? And I'm going to get to that a little bit later on. Again, any questions whatsoever, feel free to stop and, ask me. So basically, we were looking at, these modifications on the proteins, and then we became very, very interested in this one reactive carbonyl species, which is methyl glyoxal. And the reason why we selected that particular one is because it's the most potent and it is the most damaging to the endothelium. So if you take methyl glyoxal in vitro and you add it to endothelial cells in culture, it's going to kill the endothelial cells. Exactly. And I'm going to show you some movies as to exactly what is going on there. Exactly. What is also nice about this is that I could have bring in a little bit of the chemistry I learned, while I was at UV. Anyhow, so the biochemistry of this is very, very complex. So methyl glyoxal is made by several different pathways, and it is degraded by several different pathways. But this is the most complicated biochemistry slide I ever prepared. But basically what happens is it's made by many different pathways, and this is the major pathway in the cell, glyoxylase pathway that degrades it. And then this one chemical have 4 different degradation mechanisms. The reason for that is because as soon as methyl glyoxal level go up in the cell, and this pathway cannot handle it, then you have other pathways that kick in. Okay, so you have one chemical that actually is really, really, regulated in the cell.
I think that is called redox buffering.
Yeah. So it is.
Where you have one electron pair, when that is used, I think you have to
It is. Yes. So I think actually it's right. So there's a whole different class of biology where the ROS is reactive oxygen species, where you have this one electron movement. But in this particular case, what you have are the nucleophiles and the electrophiles, attacking groups. Okay. So basically, at the time, several people were showing that maybe this molecule is actually involved in the pathogenesis of diabetic complications. Okay? And people were looking at this particular target I talked to you about, and they showed that if they overexpress this particular enzyme, which will make methyl glyoxal, you can have diabetic-like complications. Other people were showing that if you overexpress glyoxylase protein, you can minimize complications. And then other people were showing, just recently, if you knock down glyoxylase, you can get diabetic-like complications. So again, this particular pathway seemed to be really important. But the problem was, methyl glyoxal is also very, very important for normal physiology. So if you had to target this for therapeutic gains, you really had to think about it. So what methyl glyoxal does is it controls anxiety in individuals. It also is very, very important for regulating non-REM sleep. Okay? So you cannot go in there and actually try to lower methyl glyoxal level in a global manner, because you're going to end up with more complications than what you started off with. So this is basically what I just said. So I had this student in my lab. He's actually a pharmacist from Saudi Arabia. So the Saudi government have a lot of students in the United States. And basically, I was talking to him and we said, if we can identify and lower the endothelial damaging subpool of methyl glyoxal, then we might be onto something. And he asked me the question, where do I look? Because we are saying that methyl glyoxal is causing a problem, but it's not all the methyl glyoxal, is only a subpool. And he did a really, really very simple experiment. Now, he's a neuroscientist. I'm a cardiovascular scientist. So when he brought his data to me, I'm looking at the data in more details because I don't know. So then basically what he was showing me is, and I'll probably, right here. So this is the blood vessels from the top of a rat brain, okay? And let me just go back. And basically what this here, this green, is a specific marker for the endothelium. So what you have is a very nice endothelium in a control brain. And in the diabetic brain, basically that endothelium is all damaged, okay? And then what he showed me is that in the diabetic brain, you had this enzyme that make methyl glyoxal called vascular adhesion protein 1. I think I talked about that earlier on, one of the enzymes. But that enzyme was not present in the endothelial cells, but it was present in the smooth muscle cells. And once we saw that, then we realized that we are onto something really, really big. So what was happening is that the smooth muscle cells, which normally respond to what the endothelial cell is kicking out, is actually creating a substance that was then working in the reverse direction to kill the endothelial cells. Okay? So again, you have to be observant. Okay, when you are doing research, you just have to pay attention to what's going on, because good data don't jump up and say, here am I, here am I. Okay, you have to know, you have to have, a little bit of background on what's going on. So then, we do a lot of hypothesis-driven research, just to make it sound, kind of good. We're not guessing, But basically what he hypothesized for his PhD thesis is that smooth muscle cells of the microvasculature is the source of the endothelial damaging MGO, and if we can lower this, then we can prevent the complications. So being a pharmacologist, we could have used genetic models to test this hypothesis, but I didn't want to use genetic models right away because these things take a lot of time to generate. There are no genetic models for what we want to do right now. So what I told him to do is let's create an adeno-associated virus and see if we can use a gene therapy strategy to quickly see if we can target these smooth muscle cells to prevent the endothelial cells from becoming dysfunctional in diabetes. So here is a rat model of type 1 diabetes, and basically what we found is that when we injected this virus into diabetic rats, this virus lower blood glucose levels. So here is the virus. This is normal diabetic animals' blood glucose high. This virus was lowering the blood glucose levels, and it was also lowering the body mass of the animals. Okay, this is a model of type 1, and then when we look at a model of type 2 diabetes, we basically saw the same thing. And this was kind of really, really exciting for us, because now we have a strategy where we can target this endothelial damaging MGO for therapeutic gains. And then he did the experiment. Now this is just like some data, but basically this took him about 6 months to do. And what he found is when he injected this virus was actually lowering this increase in vascular. So here, this is basically the gist of it. In diabetes, you have a lot of this MGO-generated enzyme being upregulated. This is an MGO marker in the blood vessels. It's difficult to actually measure MGO levels in these small microvessels, but you can use a surrogate marker. So again, it's basically just thinking about what you're doing and coming up with clues. And then when you treat with this particular virus, you can see this enzyme level in smooth muscle cell is down, and this methyl glyoxal derivative is down with treatment. Now, how it actually happens, we do not know. And in medicine, they will tell you, if you can find something that works, then you can go back and actually deal with the mechanism. If you don't have something that works and you're just rationally trying to go through, you might end up spending 50 years to actually find something. So we found something that actually worked, and then now we are going back to work out the entire mechanism by which it works out. So here is a little picture of the blood vessel on top of the brain. So what we do is we take rats and we cut out the skull on top, and then we can open up remove the skull, and then you can see the vessels. And we are very much interested in these small microvessels. They are about 25 to 50 micron in diameter. And what's nice about them is you can then, put an arrow here and an arrow here with some, specialized piece of equipment, and you can monitor how these vessels are actually dilating or contracting. Okay? So when we understand what was going on with the vessels, then we started to measure the function of the vessels. And here I'm showing you that in a control animal, the blood vessels respond very, very well to endothelial mediated relaxation, which is the key to a lot of the function of these vessels. In diabetes, actually the response is lower, and when we give this particular virus, it comes right back. Okay, so this is really, really key. We have something that actually works. This is endothelial mediated vasorelaxation, and this here is smooth muscle mediated vasorelaxation. So I'm sure you must know somebody who, when their blood pressure gets a little bit high, they take a little nitroglycerin and put it under the tongue. Basically what is happening is that nitroglycerin is causing the smooth muscle cells to relax, and as a result of that, the blood pressure is going to lower. So that's basically the same thing we are doing there for, looking at relaxation of these vesicles. So it is the endothelial mediated vasorelaxation that is compromised, but not the smooth muscle mediated vasorelaxation. And there's lots and lots of clinical data to actually show this, stuff.
So can you explain a little what you mean on this data? Because this is going to be changing dynamically, right?
Yeah.
So are you looking at the highest to the lowest, or how is this? What are these data
That's
actually?
a really, really good question. So what we have is the vessel of the rat actually exposed, and we are measuring now what is referred to as edge detection. Okay, and what it is we have this instrument that will determine the average size of the vessel.
Right, but that's not the same concept.
Well, it's going to vary plus or minus very, very. It's not that much. Actually, let's say the diameter of the vessel is 25 micron. It's going to vary 25 plus or minus 1 micron. Okay, so it's not varying by much. But if you add a compound that causes vasorelaxation, what will happen is you will see that vessel actually increasing.
So if I brought food or something,
Yeah.
and compared this when it had food source or when it was sleeping, that doesn't really change.
Well, so we are not actually looking at that sort of a thing. What we are looking at is actually so the question he asks is if you are sleeping, if there's metabolism involved.
That's got to change more than mine.
Well, in our experiments, when we do this, we anesthetize the animals because we have it here, right? And what we are doing is we are keeping it, in this anesthesia. Yeah. So if you remove the skull, it's going to be difficult not to anesthetize the animal.
Okay, so it's not a chronic opening and things awake.
No. Yeah, I think we just wanted to, quickly check and see, because again, like I mentioned earlier on, once you find something that is working, there's a rush to, do things quickly, because somebody's going to find it, and then you're in trouble.
I notice if you look at the distribution, relative percent relaxation among the 6 groups that you have there, it doesn't change even though you increase the ADP concentration tenfold. Is that what you're expecting?
So what is happening? You mean like here? Yeah. So
Yes.
So basically.
Distribution, if you look at it,
Yeah.
it's almost an identical pattern.
Yes. So what we are looking for is
Constant
actually.
identical pattern.
What we are looking at here
So
is.
that pattern didn't change.
Yes. So what we are looking at here in pharmacology, it's dose response curve. So basically, if you give a little dose, you get a small relaxation. If you give a little bit more, you get a little bit bigger relaxation.
Yeah.
But the
what I'm
errors
saying,
don't scale any
They say-
I think that's.
just
Go on.
get it right. What I'm saying is, whatever mechanism you devise to distinguish between the percentage relaxation will probably be independent of ADP.
So
That is what that is telling you, eh?
but I think what we are looking at here, so the way how the ADP works, the ADP actually binds to the endothelial cells and then and turns on nitric oxide synthase, which then makes nitric oxide, which then goes to the smooth muscle cell. So it's a sort of a sequential thing. And if you give more ADP, then you have more nitric oxide being generated. And what we are looking at here is just the response of the vessel to nitric oxide.
Yes, I know. But what I'm saying is, if you look at that, it's the same pattern. And what I tell you, what I'm
But yeah, yeah.
talking about is the relative
Right.
percent of teeth.
Right.
Now, when you write on the chemical mechanism,
Yeah.
there are mechanisms that will respond to this, and mechanisms
That are, okay.
expected
Yeah.
that pattern
I think yeah. So what we are looking at actually here, when we see the pattern remaining the same, but changing dose response manner, we are happy. Because if it's, changing when we increase the dose, then there's multiple mechanisms involved, and then that complicates the biology. Okay? So that's what we look for here. So that's a really good point, I never really thought about it like that, Okay, so then we looked at the cardiac function, and basically what we were able to see as well is that this particular virus, when we inject it, it improved cardiac function. And then this is kidney function. So everybody know somebody who have diabetes is going to end up in renal failure. And the reason why they end up in renal failure is because you have leakage of the vasculature. And this virus here can actually protect the kidney. We were really, really surprised. And then I mentioned earlier on about stroke, ischemia reperfusion injury. And this here is control. This is diabetic. And this is the virus here protecting the brain from ischemia reperfusion injury. So everything that we know that diabetes is going to cause, this particular virus was actually blunting it or preventing it. Okay? And then we wanted to make sure that this particular virus was not causing adverse cognitive impairment in these animals, because that's it's working well to fix the kidney, fix the heart, fix whatever, but then if it messes up the brain, then that's a little bit of a problem. So again, my student decided he was going to do a very quick and dirty experiment. So a few years back in Nature Protocols, this guy came up with an experiment where if you put a little piece of cotton in the cage of a rat or a mouse, that rat overnight is going to shred that cotton and build a nest in the middle of the cage. That's an innate response. Okay, so that means everything is functioning well. Now that movement requires you to think a little bit. It requires your mouth to be able to gnaw, and it requires your foot to be, moving and your, whatever and stuff like that. So basically, this simple assay is telling you whether the animal is functioning normally. So it's a daily living function. Okay, so again, you don't have to think very, very complex, right? And what he found is that in control animal, it builds a nest. In a diabetic animal, they don't even move the cotton. And if you give a blank virus, a control virus that doesn't have the gene, it doesn't even work. And then if you give the insulin, it works well because you're now reversing pretty much everything. And if you give the virus, it's working very well. But what he also observed is that this little wet thing in the cage here, if you are diabetic, you tend to urinate a lot. And he realized that these animals were not urinating as much as the diabetic animals. So again, it was attesting to this virus was actually protecting renal function. Okay? So again, it's some very, very simple experiments that you can do, to get to the mechanism. And then what I told him to do, which is a very, very elaborate experiment. I said, okay, what I want to see now is actually what is going on in the brain of these animals. So he, generated some animals, and then he started to dice and slice the brain of these animals, and we were starting to look for leakage, exactly what is going on. So and this is clinically relevant. Many individuals who have diabetes, they have cerebral vascular leakage, or they have cerebral blockage of the vessels. And this is a normal animal, and what you see, this is a blood vessel here. You see no leakage. This is a blood vessel, cut sagittal. This is a coronal. This is a blood vessel called cut sagittal. No leakage, not whatsoever. But if you just look at the diabetic animals, then you start to see leakage. This here is the hippocampus of the rat that is involved in memory. This here is the thalamus. Oh, let me just back up. This here is the thalamus, like this region here. This region is the cerebral cortex. So what we basically started to see was this spotted leakage throughout the entire brain of the animal. Yeah. So good, really good question. So a friend of mine actually is working on the eye, and basically you see you see the same thing here. You see the same sort of phenotype. But we were just focusing. So I think we had a couple of papers earlier this year talking about what is going on in the eye and diabetes. So what you see is this leakage, and then when we give this virus was able to prevent all of this leakage that is occurring in the brain. So then we wanted to know, well, exactly what is the mechanism by which this thing is actually causing this problem. What's that? Yeah, so I showed you the data early on where we restore the integrity of the vessels.
You can't restore it.
Yes, because so this here is basically what happens. The reason why we have this leakage that I just showed you is because the endothelial cells actually forms this protective barrier. And if you kill all the endothelial cells, you will just have a space for the blood to actually enter into the parenchyma of the brain and any of the other organs. But if you have the endothelial cells, then you wouldn't have the leakage occurring.
But you can't, once those endothelial cells
They're gone.
There,
Yeah. So I work also with a clinician, and what we do is we isolate endothelial progenitor cells from patients. And basically we see the same thing occurring. In the endothelial lining, it's an equilibrium between endothelial progenitor cells, that is new cells coming in to replace the dying cells. But if you have the new cells that are coming in compromised, then once these cells up here are gone, then there's no new cells to actually come in and replace it. Any questions there again? So that's basically the gist of it, what we found. And then whenever you see, this sort of a leakage, you always want to make sure that you know what you're talking about. So if there is blood, there's going to be a protein called IgG. You just have to make sure that you have some of the blood markers and things like that. But these are some really, really nice images, because I had this guy look at hundreds of these to pick out these nice ones to show here today. Anyhow, but when you have also leakage of blood into the brain parenchyma, what you end up doing is actually increasing inflammation. And a good mark of inflammation is activation of astrocytes. And here, when astrocytes become activated, they change shape. They become very star-shaped. When they are not activated, you just see like dots in the brain. Okay, and when they become activated, you see these nice star-shaped like features. And then when we treat with the virus, because we are not causing leakage, you can actually prevent the astrocyte activation. And one of the things that we found is this astrocyte activation was primarily around the site of leakage. And then, just to make sure that everything is working well, I am an electrophysiologist by training as well. We take some hippocampal slices and we stick an electrode here and stick a next one here, and then we can measure how well neural transmission is occurring. So basically, this is just a graph showing that neural transmission is compromised in the diabetic animal, but we can restore it when we treat with the virus. So basically, now we know that everything is working well. I asked the student, just go and measure metyl-glyoxal level in the brain to make sure that it is upregulated in diabetes and this virus is low end. So here he came back with this data. It's increased almost 5-fold in the brain of diabetic animals. When we treat with the virus, it's actually coming way back down. So now we know that metyl-glyoxal is actually involved in the whole process.
So the question I have, and the results are...
No,
I'm really
go ahead.
enjoying this.
Yeah.
What I'm curious about is in the biochemistry and in the metabolic pathway, why is that regulated?
Why is this upregulated? So metyl-glyoxal is upregulated.
In diabetics.
Yeah. So what you have is metyl-glyoxal is generated spontaneously from glucose oxidation. There are quite a few enzymes as well that actually operate when they produce metyl-glyoxal as a byproduct of their degradation reaction.
Okay,
So all
you
of
had the
them.
metabolic network, and it wasn't quite apparent where in the metabolic
Okay. So
network.
yeah. So
That's right. We had to go into
Okay,
detail
yeah.
on that, but I
Yeah,
really
so
curious.
all of the metabolic pathways that produce metyl-glyoxal is upregulated in diabetes. You have increased glucose metabolism, you have increased lipid metabolism, you have increased expression of enzymes like VAP1, you have increased polyol flux. Everything that is actually generating metyl-glyoxal is upregulated in diabetes.
And the fluorescence.
What's that? Yeah,
And the
and the fluorescence.
Yeah,
Yeah. So then, whenever you measure methyl glyoxal activity, you always have to measure, activity of one of the enzymes. And basically, my student just measured the activity of glyoxylase, and it's down. So this is the enzyme that breaks down methyl glyoxal, the primary pathway. It is down-regulated in diabetes. And then when we treat with the virus, it is upregulated. And the reason why I'm showing all of these extra data is because when you are trying to say something, you have to have the right set of controls. Every control must be there, because people are going to say, well, I didn't see this, I didn't see that, etc., etc. So you have to have the right control. And this is getting back to the question that Dr. Narayan asked. This is methyl glyoxal that is sold commercially. It's yellow. So there's a lot of preservatives in it. And we had to synthesize it in the lab to make sure that we were using pure stuff. And this, again, chemistry background, we were able to synthesize it very, very quickly. And then the question we asked is, will this thing kill the various cell types? And what we find is that endothelial cells, brain human microvascular endothelial cells, are very, very susceptible to methyl glyoxal. Smooth muscle cells and neurons are not big. And the reason for that is because endothelial cells doesn't have that enzyme that breaks down methyl glyoxal. Only the smooth muscle cells have a lot of it, and the neurons have a lot of it. And then mechanism that you talked about, why is methyl glyoxal actually killing these cells? Well, what it does, like you mentioned, it's producing a lot of superoxide in these cells. And it's doing it via the mitochondria, which is the energy supply of the cell. And here I have a nice little movie to actually show. So this is mitochondrial labeling. And the green is a mitochondrial dye, and the red or orange is actually a dye that we use whenever there's a change in superoxide generated in the cell. And this is just like a 10-second movie. Well, not even so much. But what you see is as soon as you add methyl glyoxal, these cells just start to glow. And this is with cardiac myocytes, but we see it with all the various cell types that we can actually think about. So now we know exactly how it is methyl glyoxal is actually killing the cells. And then we can go in a little bit further and talk about the various complexes in the electron transport chain that are involved in the process. But I don't think, I want to go in here. We know every protein that is involved in this whole, process. Again, this is the cardiac progenitor cells, and we basically see the same thing here that is occurring. And then I didn't want to stop there. I told my student, just go and isolate mitochondria, and let's check and make sure that the mitochondria is actually what is causing the problem. So we isolate mitochondria, and just to make sure that the purity of the mitochondria was good enough, we do EM. Okay, so here you have nice mitochondria, and they look very nice. And then we use a next technique to actually show that when you add methyl glyoxal to the mitochondria itself, you have this increase in ROS production. So basically, we know a lot about what we didn't know about, how it is methyl glyoxal is actually causing this problem. But what was unique about our work is that we were able for the first time to identify a subpool that we can then target for therapeutic gains. And the reason why we were able to do that is because I was looking at everything not from a biologist standpoint, but I was looking at it from a chemist standpoint as well. I was bringing the biology and the chemistry together to actually solve problems. Okay, so in summary, basically, this is my summary slide. I don't have to go back and tell you what I find. As a first student, think about the problem at hand. Talk to people. I used to talk to a lot of people, right? I used to go to Dr. Narayan Singh. I used to go to Dr. Stout. I used to talk to everybody I'm talking to, because everybody has a different viewpoint. If you don't talk, then you wouldn't get a different viewpoint. My second point, pay attention to your data. When you get your data, look at your data. Think about what your data is telling you. And luck always increases for the prepared mind. Okay? Familiarize yourself with techniques. So if you have a problem at hand, don't say, I have a salad, I'm going to be putting ranch dressing, blue cheese, next one. Basically, you're doing the same thing, right? If you want to solve a problem, you have to bring in a whole bunch of techniques. Be multidisciplinary. That's what Dr. Stout was telling me here. The programs here are basically looking at things from different angles. And the last point is actually read around your topic. Not because you are doing, let's say, something in ecology or whatever, that doesn't mean to say you can't read something on biology or history or whatever. Read as broad as possible, because you're going to get points that will help you later on. And then finally, this is my institution here, University of Nebraska Medical Center. I am on the 3rd floor. I think I have 2 of these modules, 2 of these lights there. And then again, like I mentioned, this is part of my group here, and I ask a lot of people, for advice. I want to point out Dr. Jai Paulson, because actually he used to be, well, I think he still is, Dr. Chanyan high school classmate, and he's now one of my mentors. So again, the moral of the story there is you never know who you're going to meet in life to help and guide you as you are going along. Right? So I hope there are more questions, and I'm free to answer anything that you guys want to talk about with respect to this book. Mm-hmm. Mm-hmm. Bring it down to a low level, then kind of get into.
Why would you have called it a subfood? Simply because it existed in one part?
No. So what we want, in, yeah.
Like if you have this compound, if you have NG,
but this is a really, really good question. So the clinicians, they measure methyl glyoxal in the blood. And the concentration of methyl glyoxal they measure is about 1 micromolar. So in healthy individuals, what you have is 500 nanomolar. In a diabetic individual, you may have 1 or 2 micromolar. But if you take 1 or 2 micromolar and you put it on cells, it does nothing. Right? So basically, you have now a concentration that is doing nothing. And the clinicians are really excited about this, but they were never able to show severity. Well, so we kind of, just wanted to find this endothelial damaging pool. And that's what we were able to actually identify in this particular, set of studies. And now the other thing that I didn't mention as well is when people are actually developing strategies for gene therapy, what they do is they create a gene with an adeno-associated virus that go in every cell type, right? What we wanted to do is actually let this protein that we are interested in express in the bad cells, and if a cell is not defective, this protein will not express. So what we end up doing is actually not just making an adeno-associated virus, but I use various protein-specific promoters to regulate it. And that concept have never been, demonstrated before. So if a cell is good, then this protein will not express. If the cell is bad, then the protein will express. But if that bad cell gets back good, then you will shut down expression of the protein. So we were now able to actually regulate how this protein is moving back and forth and back and forth. And our institution got really excited about this because it was never demonstrated, in this particular manner, before. So some of the details I'm leaving out because I think they are not, that important. But as you ask me questions, I'm going to explain. Repeat one more time.
From the results that you're getting, very first results that you're getting from your research, from your point of view, would you consider research in insulin mimesis dying or dead?
Research?
Insulin mimesis. Insulin mimesis.
That's a tough one to answer. Yeah, it's really.
What are your consideratory shortcomings?
So I have actually, so let me tell you how I look at it, right? I have a, I have a passion for research, right? I like research, for me, right? That's just for me, right? And to me, it's, I get up in the morning, I'm showering, and I'm thinking, I'm just thinking, what am I doing? What am I, thinking of, So for me, I get up in the morning and I'm just, I am excited, whatever, and I try to impart that excitement on my students, right? If you come in the morning and you're looking sour and you don't want to do something, I tell you, go home, because I don't want you, yeah. No, because it's better, because you might just, so for me, it's, what the problem we have right now in the US is that the funding is cut. And people have to come up with creative ways to get money to do research, right? And like I am in heart and lung, and my grants used to get funded at, let's say, 12%. Now, The funding is 8% and 6% in my institution. Now, I got a grant recently that got a score of 15 percentile. And that grant didn't even make it to the review section. You know what I mean? So what I end up doing is I am actually funded by, I got grants from I got an innovation grant recently for a huge amount of money that is funding this work. I got grants, small grants to generate animal models. I, spoke to my chairman and I convinced him that what I am doing is good, and then he, throws some money by me. I have grants pending from the Ayacoca Foundation. I have grants pending at NIH. So again, all it means is you have to write more. You know what I mean? And you have to sell your science. Yeah.
Inspire you to go in this direction and not because a lot of work is still being done in that. For someone who is basically, I would say,
yeah, go ahead.
some distance yet. I have a student working on two potential families right now, drugs.
Mm-hmm.
What, from your experience, rather than those with other professionals in that area, what are the points of concern that you like?
Oh, so that's to me, that is a negative approach. Points of when you start to talk about points of concern. I don't worry about that, right? It's yeah, it's yeah. So, yeah. Yeah, so the difference actually there is exogenous versus endogenous, right? So the mimetics actually is exogenous, right? And there's a guy up in University of British Columbia, John McNeil, who has tried pretty much everything, right, when it comes to exogenous molecules, and none of them have actually made it to the clinics. He's tried, in addition to what you are talking about, a whole bunch of vanadium compounds, etc., etc., etc. And to me, those vanadium compounds, they're not really going to solve the problem, because what they are trying to do, and I think you kind of missed the focus of my point, I don't want to make a next glucose-lowering drugs. What I want to do is to make a drug that can reduce complications, because we have so much glucose-lowering drugs. Recently, this last year, I think one came out that actually works on the renal sodium or renal glucose transporter, right? But the problem is that individuals with diabetes mellitus, they don't really have a problem actually taking drugs. It's when the drugs don't work, then they give up. And what I wanted to do here, and I think I said it in the second or third slide, is we wanted to create an adjunct to glucose-lowering drugs that could reduce the complications. See what I mean? Not necessarily a glucose-lowering drug, because glucose-lowering is not the problem. We have tons and tons of drugs, but we wanted to come up with something that can actually reduce the complications, the end organ complications. Mm-hmm. Right. Mm-hmm.
And according to your
yeah. Mm-hmm.
that is due to metabolism of the triglycerides.
Yeah, several sources.
So there's a general
Mm-hmm.
question. So if somebody is diabetic
Uh-huh.
And they're on medication,
Mm-hmm.
point of let's say metformin,
Yeah.
so increase insulin sensitivity.
Yeah. Mm-hmm. I'm listening.
50 meters is causing problems, and that is as a result of increased metabolism of fats and sugars. Then it's almost as if the drugs that the diabetics are taking is causing.
Well, it's not necessarily the drugs that they are taking. Yeah. So let me put it in a nutshell, right? When you are diabetic, let's say this is your blood glucose levels here when you are healthy. And you're diabetic, your blood glucose levels go up a little bit. But even if you take drugs, you don't want your blood glucose levels to come back down to normal, Because if it come too close to normal, it could drop below normal, and then you end up with hypoglycemia, which is a problem. And hypoglycemia is an immediate, your heart starts to beat fast, you start a cold sweat, etc. So what most of the drugs actually do is instead of bringing the glucose levels back to normal, they bring it a little bit above normal.
So you're saying that
That slight increase is sufficient to actually increase production of methyl glycol.
Right, so that's what I'm saying, that the drugs.
Well, it's not the drugs.
But the treatment.
Well, yeah, so the treatment actually is not a perfect treatment. And even if you take it as well, even the pumps that they use, it cannot come back down to normal.
Because I'm trying to rationalize this versus
Yeah.
metformin versus
So the really good, so diet and exercise cure everything, right? But it doesn't cure this, right? Yeah, so
You're forgetting something else here. You have a very, very complicated mechanism. You have several products that are formed. Now, we don't know what is happening when glucose is lower. When glucose is lower, if you have competitive reactions among those things, the amount of NGO that is formed may not drop comparatively. It's not linear.
That's actually what happens. So actually, that is exactly what happens. So when you lower blood glucose levels, for reasons that we do not know, methyl glyoxide production is still elevated. So you shift metabolism.
The reason is you have to draw a competitive mechanism for that. That gives you a clue to the kind of mechanism you have to draw.
So it is...
glucose lowering wouldn't help because the ultimate problem is the damage of
the vessels, is the damage of the cells.
And that is still around.
And if you have that, and what we are showing here is the smooth muscle cells, production of MGO is just upregulated. So again, sometimes, and I used to think, pretty much just like how you were thinking. But what I realized that sometimes you need, if you find a cure or you find something that works, then you go back and deal with the mechanism. Don't worry about that first, and it's very, very difficult to actually, find something that is, working along those lines.
But you see, all of those things that you went through, and kudos to you, because you obeyed all of the tenets of good research, in that you eliminated every confounder. Each one of those will give you a clue as to the kind of mechanism that you ultimately have to. And it looks like a very complicated mechanism because you're doing it longer rates. But you might be able to guess at the kind of balance in the rates without knowing the actual rate constants.
So,
then you can draw the mechanism.
Okay, thank you very much.
I have a portion I got to teach.
Okay, so I think the one of the things is that there's never going to be a perfect cure for any disease. What our ultimate goal is, actually, we don't mind if you have end organ complications when you are 85, 90, but we don't want you as a diabetic to start having end organ complications at 50 or at 45 or at 60. That's what we are trying to.
I have to go, but the end result of all this is the therapeutic intervention for people with diabetes mellitus. What would it be? Gene therapy?
Well, what we are so there are many options, right? But what we are trying to do is we are trying to develop maybe once every 6-month injection. You take it in addition to your glucose lowering drugs plus your insulin to just reduce end organ complications.
Okay, because the next question I was going to ask is whether the therapeutic solution makes metformin and all the other drugs, whether superfluous or obsolete.
Well, it would. So it would not. So that's the point I was trying to make earlier on. Metformin, and you know, a lot of people know this, right? Metformin is good for 6 years, and then after metformin, then you have to go on to maybe sulfonylureas plus, Pargelol or one of these insulin, analogs. So what we have right now is drugs that are good only for a short period of time. And eventually what you're going to end up with if you are diabetic are these end organ complications. Even though you are taking all of these drugs within a short period of time, 15 to 20 years. And what we are trying to do is we are trying to come up with the next strategy that could probably not have you having end organ complications in 15 years, but if you can have end organ complications in 45 years, that is much better. You see?
And the third question I want to ask,
No, good.
anticipated time horizon with regard to, I imagine that you are doing clinical trials and that sort of thing?
It's all about the money. It's all about the money. So the first thing you have to do, so we have the patent application pending. Once the patent application is approved, which most likely will, I heard from the lawyers recently, then it's going to be, we're going to attempt to commercialize it and get a manufacturer to actually, or a company to be interested in it. And that's where the marketing aspect of science actually come in. So that's where I have to go there and sell ice to an Eskimo. And then, once they pick it up, then you actually, you have to convince them. But what most companies are doing now, they don't want to have the risk of identifying a compound. They want like institutions like ours and investigators like me to identify it first, and then they just piggyback onto something that they know is working.
But it would be nice if you could tie macular degeneration.
Well, I think, so I am working, so we have a couple of papers, I think, that came out this year, not specifically looking at macular degeneration, but, age-related,
So basically, the University of Nebraska is in terms of the commercialization of research,
Yeah.
right? I presume that all are going to reap the benefits of it. You're going to commercialize it.
So it's 30.
Very well.
So the investigator gets 30, wanted. The investigator gets wanted, the department gets wanted, and the university takes wanted. So that's how it's done. Yeah, that's how it's done. So it's Yeah. So every university. Yeah.